I'm going to talk about the studies from our lab that combine big data for personalized medicine in a particular field of breast cancer. Most of the work was carried out by Avital Gilham, a PhD student in the lab, and by a postdoc, Daphne Weisskos. So when talking about personalized medicine, we usually refer to looking at the DNA of one particular individual, comparing it to another individual, or to a large database, in order to understand the differences between the DNA. And then we want to refer to a particular therapy or change of lifestyle or treatment or drug that can help that particular individual. I want to show that in the lab we combine different fields for personalized medicine. So we can combine DNA and RNA information. We combine specific tissue and surrogate tissue. You can't always reach the brain in order to study it. You can't always reach a tumor to study it. So you have to look further downstream. And there's a lot of big data out there. There's a lot of information. We gain a lot of information, but eventually we want to narrow down to 1, 2, or 3 genes that we want to treat. So we look at all these levels when looking at personalized medicine, and we combine them all in our studies in the lab. This is the central dogma of gene expression up to about 10 or 15 years ago. It was quite simple, but since then we are gaining a lot of information about RNA in the cell. So we have a lot of RNA in the cell. The most popular one, or known one, is our microRNA, small RNA that would bind an mRNA and shut off its expression, but there are many other RNAs in the cell. So our cell is a very large bag of RNAs. There's a small amount of DNA and also proteins around it. This is how we see it. If we look at our DNA, only about 3% of the DNA codes for protein. However, 70% of the DNA is expressed as RNA. Now we can take the RNA out of the cell, we can sequence it using NGS technology, and we can see that 70% of the DNA in a particular cell or time point could be expressed. So this might be a terrible evolutionary mistake, or maybe there's some meaning into it. So we're taking these RNAs, we're trying to understand it, what they do in the cell. We should all thank Fred Sanger, a professor from Cambridge, UK, who passed away about 3 years ago at the age of 95, one of the only ones who received 2 Nobel Prizes for using the inherent properties of nucleotides binding to each other. So he took a G, and he saw that it bound to a C, so he figured out that the C is the missing link in the unknown DNA. And we use machines that basically use the same properties today. The first generation sequencer here on top used the same idea, only automated, and also the second generation sequencer used these properties of binding of the nucleotides. At the beginning, these automated machines read only about 96 reactions or 100 reactions. Now we read millions of reactions. And in my lecture, I usually compare the first generation sequencer to a car from about 100 years ago. Today, we're flying on a spaceship orbiting Earth about 10,000 times faster than that car. The leap in our ability to read DNA is about 100,000 times more. From the first generation to the second generation, and the evolution of machine doesn't stop there. What we use in the lab now is the small machine you can see here. This is the MinION from Oxford Nanopore Technology. We're sequencing DNA in our lab using this machine, and I'm running out of metaphors, but I think we'll be riding a meteor soon and reading even more information. A lot of information, and obviously we want to ask, what do we want to do with all this data? So how do we combine the different fields? How do we combine DNA and RNA? We used to see the entire DNA from beginning to end in images that look like this. But obviously, this is like looking at planet Earth from space. So you can see the different continents. You can see where they are, but you can't really look at the specific nucleotide of each DNA. And now with NGS technology, we could really zoom into the continents, to the different countries, to the cities, even to a particular street. We know we could do that using Google Maps and Google Earth, and a particular house, and even look at a particular window. So this is what we do today. We could read the human DNA from beginning to end. And this is how we look at it. This is how we view it. So we have the entire variance at the outer circle. You can even leave this room, I can go to one of your seats, I can take a piece of paper, I can wipe the table, and I will have your DNA. And a week later, I can have the entire sequences of your DNA from beginning to end. And each of us has a few hundreds of thousands of nucleotides different when we compare it to the database. This is the outer circle, all the variants, and of course we have to apply different filters. We have many polymorphisms, many mutations, but we want to reach the ones that are significant. So from left to right, you can see from the outer circles, we're moving from inside exons, missense mutations, and nonsense mutations. This is from left to right. And on the right-hand side circles, we're moving from frequency in the population, allele frequency, above 5% or lower than 5%, lower than 1%, and novel ones. In the center, in the overlap, here we will have nonsense mutations, which are novel, which are unique for you, for each individual. And when we sequence an individual, we can find about 20 or 30 novel mutations that seem to be detrimental, seem to have some effect on the protein structure, on the protein coding. Many times we don't know what it means, but we're trying to understand. The more we sequence, the more we understand better the DNA and the effect on the proteins of each and every one of you. We sequenced in the past few years about 1,000 individuals here in Israel. So we're looking at the DNA, and obviously this applies to the RNA and to the proteins. We're also looking at big data. We're looking at a lot of information out there, but eventually, as I mentioned before, we want to reach 1, 2, 3 genes which are significant. So this is the approach we took. We're looking at micronases, oncogene regulators. Again, micronases are small RNAs, about 20 nucleotides long. They float around their cells. We have thousands of copies of them. They can bind an mRNA, and once they bind an mRNA, they cause its degradation or they inhibit translation. So they shut off expression. This is the data sets we took. The first data sets we took is a list of mutations. This could be a mutations from an individual. This can also be a database of polymorphisms, SNP, single nucleotide polymorphisms. Polymorphisms are known changes in the DNA which are prevalent at about 1% or more. That's the top circle. On the left-hand side, you can see that we have a variable list of genes. Over here, we took oncogenes due to the interest in this particular study. And the last circle at the bottom right-hand side are microRNA targets. We can look at a microRNA and predict its binding site. So we know, we can predict at high probability, if it's a highly conserved binding site, we can assume that this microRNA would bind there and cause degradation of that gene. So here in the center, we will receive changes in the population, polymorphisms, that happen to land in very interesting genes, in oncogenes, that happen to land in a microRNA binding site. So this particular site in an oncogene could either bind or not bind a microRNA. So this is the list that we generated. We received many genes there. In the particular study I will mention here, we looked at breast cancer metastasis. So we think it's very relevant to study breast cancer. We all know that 1 in 8 women will be diagnosed with breast cancer throughout their lifetime. There are many studies trying to understand and alleviate the suffering of breast cancer patients. We know that if you can self-restrain or if you can keep the primary tumor in one location and prevent the metastasis, this would be a very important advancement in treating breast cancer. Sometimes some physicians might say that you can even leave a primary tumor and not treat it. And despite the millions and billions of dollars invested in research in breast cancer, we know that there's only a slight increase in overall survival rate. Also, it's very important to mention that due to widescreen mammography programs, early detection is becoming more prevalent. So this is what we wanted to address. And now I want to show you the functional screens we performed based on the 3 circles I showed you at the beginning. So we took all the genes associated with breast cancer. This is a very wide filter. We have about 4,000 genes here. Also those that are mentioned in the literature to be associated with breast cancer. Then we took from this group ones that have a microRNA binding site at high probability, highly conserved microRNA binding site. It is known that about 50% of the genes have a microRNA binding site, and here you can see that we have about 2,600 of them have a microRNA binding site. Then another filter we looked at is for those particular sites, which one has a polymorphism, a change in the DNA that could either cause a binding or abrogation or removal of the binding by a change of one nucleotide. Obviously, this is just computational at this stage. And since we wanted to address the metastasis stage, the movement from the primary tumor to a secondary tumor, our final filter was looking at genes with cytoskeleton organization or associated with cytoskeleton organization. Again, for a tumor to move from one side to the other, it has to change the cytoskeleton, squeeze through the bloodstream, and then squeeze again at the secondary side and be seeded again. So here we received 19 genes, very interesting genes. About 1/3 of them are known to be associated with metastasis in breast cancer or other types of cancer, and others are less known. So we chose one of these genes as a case study. We chose paladin. It's marked there in the center. It is less known. There are very few papers about it, but it looked very interesting. So why paladin? Paladin increases in invasive cancer cells. It has a very distinct feature. It's a component of the actin cytoskeleton, forms microfilaments and controls adhesion and contraction. Also, it's part of the podosomes, kind of a finger-like structures that allows the movement of the cells from one side to the other. First of all, I wanted to show you the polymorphism. You can see in one DNA there's a G, in the other DNA there's a C. It happens to land at the critical site of two micronases, MIR182 and MIR96. So potentially change of one nucleotide wouldn't allow the close of this zipper between the microRNA and the mRNA. And if you don't close this zipper, you wouldn't get degradation. At least at this stage, it's only computational. So obviously the first step we want to see is that this is indeed true. And we perform what is called a luciferase assay, which allows the measurement, the accurate measurement of the level of a protein. Here on the left-hand side, you can see that by changing in the paladin nucleotide G to nucleotide C, you see a downregulation, so we remove the binding of the microRNA by a change of one nucleotide, and then you have, works the other way around. When you have the binding, you have downregulation with the C. When you change it to a C, you remove the microRNA, and then you have upregulation of the gene. This works in various cell lines. This works for the two microRNAs we identified. This works on the RNA level, on the protein level. This works on constitutive expression and transient expression. So we were confident to move to the next stage. In the next stage, we looked at the functional effects. So we were changing the levels of the microRNAs, and we were checking cancerous features of this DNA, of these cells. Here you can see a scratch assay where we increase the level of MIR182 or MIR96. We downregulate the level of paladin, and then you have a slower closure in the scratch assay. And this also worked in the invasion assay and then migration assay below, and we have the quantitation on the right-hand side. So this was in vitro, it was encouraging, and then we moved to experiments in vivo. What Avital generated is a triple negative cancer cell line expressing M. cherry, a red color that we can follow these cells in vivo. So the experiment was like this. At day 0, we took the female mice, we injected the cells in a mammary pad, and then 2 weeks later, when it came to our clinic, we told them to identify a tumor, a very large tumor. It was about half a centimeter in size. We removed the tumor. This is day 13 here. And then we sent them back home, and we wished them good luck. We told them that the operation was very successful. There were 3 types of cells. One of them was constitutively expressing a scrambled RNA. This was our control. And the other 2, one of them was expressing MIR182, and the third one was expressing MIR96. So about 20 days later, we brought them back into our lab, and we performed 3 experiments. One of them is a micro-CT to see whether there's metastasis in these mice. The other one was with a Maestro machine, where you can see the red M. cherry cells, whether they moved around the body. And the third one was removing the various organs and using a pathologist to look to see whether there are any tumors in the different organs. So here I can show you that we haven't affected the primary tumor. I want to remind you that our goal is metastasis and not the primary tumor. You can see that the tumors here are roughly the same size. Here on the right-hand side, you can see that the tumor volumes were roughly the same. This is the primary tumor. We haven't affected it. And at the bottom, you can see the mice weight. The weight of the mice was roughly the same in the experiment and the control. And here are some of the results. Here you can see the removal of the lungs. These are images of the Maestro machine. On the top row, you can see 6 mice, and in all of them, you can see metastasis in the lung. This is a secondary site following injection of the primary tumor into the mammary pad. Below that, you can see the lungs of the primary tumors that expressed either MIR182 or MIR96. Besides one probably artificial expression here, you can see that there's no metastasis in the lungs after injection of the primary tumor. This is 1 month after injection, about 20 days after removal of the primary tumor. So this was very encouraging. You can see on the right-hand side the quantitation. That means that tumors that will constitutively express MIR182 or MIR96 can hardly or maybe not at all move from one site to the other, probably due to reduction of several proteins. What we showed that pallidin went down. So at this stage you can say, okay, this is a little bit cheating because you're using tumor cells that you increase the levels of one of the microRNAs. Can we mimic something that really happens in patients? So this led us to our third experiment. This is another experiment in vivo. Here we were helped by a colleague of mine at MIT, Dr. Nathalie Arzi, who's a chemist there, who generated nanoparticles for our experiment. And the experiment started the same way. So we took female mice. We injected now naive breast cancer, triple-negative breast cancer cells into the mammary pad. 2 weeks later, the mice came to our clinic. We identified the tumor of these naive cells. They were also marked by M-cherry. The tumors were about half a centimeter in size, which is quite large for a mouse. At this point, we injected a hydrogel containing nanoparticles into the microenvironment of the tumor, so at several locations around the tumor. These hydrogels had nanoparticles that slowly released 2 microRNAs, MIR182 and MIR96. And as a control, we used the scrambled one that expressed scrambled RNA as a control. 48 hours after the injection of the hydrogel, we performed the experiment, removal of the primary tumor. The hydrogel was still in that area and still releasing MIR96 and 182 under control. 20 days after that, then we sent the mice home and we wished them good luck. So about 20 days later, the mice came back into the clinic. And here we performed again 3 experiments. One of them is a micro-CT to look at metastasis. The second experiment was a Maestro, so you can see the metastatic site because these cells were expressing M-cherry. And the third one was removal of the organs and sending it to a pathological level. These are the nanoparticles. We tried different types of nanoparticles. Besides releasing our microRNAs, we also tried different combinations of drugs. So here you can see this one also expressed cisplatin. Our cells are marked red, so the nanoparticles are marked green, so we can follow them. Here you can see some of the mice. Here you can see without a drug, so these are just the nanoparticles expressing our 2 microRNAs in our control. You can see on the top right-hand side, MIR96 in red and cherry. So here you can see the tumor, primary tumor. Here you can see in green the nanoparticles. So they're surrounding the tumor and they also enter the tumor. Below that you can see the MIR182 one and you can see the control one, and these are the tumors. We haven't affected the primary tumor. This was not our goal. Here at the bottom you can see that when we add different combinations of drugs, over here in the image you can see that cisplatin, we also affected the primary tumor, which was not our goal, but you can see we can also reduce the size of the primary tumor. And this is without the nanoparticles. So 20 days later, after removal of the tumor, 20 days plus 2 days after injection of the nanoparticles that released MIR96 and 182, we took these animals to the micro-CT. What you see here is a cross-section of a representative mouse of our control group. This was the one that was releasing a scrambled RNA. Everywhere you see a red arrow, here in red, this is a metastatic site in the lungs. Metastatic from the primary tumor we generated in the breast cancer. The image above that, I will just increase it in size a bit, is our experiment. So the image you're about to see next is nothing less than beautiful. It is the exact flash moment where everything came together. So years of work, months of experiments, repeating the experiments over and over and over again, late nights, over the weekend experiments, mostly of Avital, who came and has done all these experiments. This is the moment where everything came together, and this is the moment that probably a scientist lives for. We have stopped the spread of breast cancer in mice, triple-negative breast cancer, and here you can see that we have hardly any red arrows, meaning that we have hardly any metastatic sites when expressing the 2 microRNAs around the primary tumor. We also looked at other organs. We looked at the liver, we looked at these are the lungs, we looked at the liver, we looked at the brain, we looked at the spleen. We haven't seen any metastatic sites 20 days later after expressing these microRNAs. In order to show that this might be effective in patients as well, so what Daphne did, she downloaded the TCGA database, which is a database of clinical samples from the NCBI. And what we wanted to see here is at least an anti-correlation of the microRNAs and pallidin in the tumors itself. We saw that in breast cancer. So what you can see here on the right-hand side, this is a very nice correlation between miR-96 and 182. You can see a high correlation between the 2 microRNAs, meaning that they are expressed together. And here you can see a very slight but significant anti-correlation between pallidin and miR-182 and pallidin and miR-96. So this means that in real patients, you can see an anti-correlation of the microRNA, and now we're trying to associate these anti-correlation to clinical data as well. I can also mention that we also have another list of genes we're looking at. We're looking at particular pathways, and we're also looking at different types of cancer. We can see that this anti-correlation between the genes we identified and the microRNAs might be even stronger in other types of cancer. So we believe we have a system now to scan a particular mutation of a particular individual. It could be a large database, but it can also be a very particular individual we sequence. And we look at the 3' UTR. We might even think that this is kind of a missing heritability. Everybody looks at the coding sequences, and we are now shifting the focus from the coding sequences to the 3' UTRs, and we can see that there's a power there, changing nucleotides, changing mutations, changing the sequence in the 3' UTR could really have a significant effect in leading to breast cancer metastases. So once we have candidate, we take it to step 2. In step 2, we check it in vitro to see that indeed changing one nucleotide could release the microRNA and upregulate the gene. Then we look at large databases such as the TCGA, and then we take it for experiments in vivo in mice. Summarize this experiment, crossing a large data set can lead to genes of interest, specific genes of interest. Combining DNA and RNA information is very, very relevant for regulating cancer, and personalized medicine might be effective for breast cancer, and this might be the first initial step that might prove it. This is the team that performs all the experiments. Most of the studies were carried out by Avital here. Daphne Weissglass did all the bioinformatics. And I thank you for your attention.
I don't know enough about microRNA to know if this is a silly question or not, but this seems to be very good for diagnostics or prognosis, but is this also relevant as a therapeutic? Is that what you're...
So we are trying, yes, we're trying to show that this is also effective for therapeutic. Potentially, if you identify the cancer at an early stage and you could reach the site and release these microRNAs, you can change the properties of the primary tumor, and yes, you can prevent it. There are many issues with delivery. we usually don't deal with delivery. We outsource the delivery project to another lab. Yes, but we have proved here that at least if you could reach the exact site and release the proper microRNAs, it would have a dominant effect. And it's not a silly question.
Keeping this strand of mind, this thinking, it means that the patient would have to get constantly this microRNA to stop the metastasis?
Okay, so that's a good question. How would we eventually treat the patient? We try not to work on delivery. There's a very large lab working on delivery and bringing the exact microRNAs to the exact site. We haven't tried a long-term study yet. We are planning to do it. So we don't know how effective it is more than 1 month, but we are planning to do it, and I don't have a reply for that at the moment.
Related to that question, are there any thoughts of maybe a CRISPR type of therapy that would swap out the problematic SNP and then it's a one-time?
Okay, so this is a germline mutation. This is very important. That means you're born with that. CRISPR type-like mutation probably has to first undergo an ethical approval, which is an issue by itself. And once CRISPR would be approved for other experiments, I assume you could also do it here. I heard there's a study in China now removing, I think, lymphocytes from the blood of cancer patients, modifying them, reinserting them into the cancer patient. So it's done outside the body, not inside the body.
I was thinking local delivery.
Local delivery for CRISPR, well, as long as it could be self-contained enough, yes.